Thank you, Kevin.
Okay.
That was amazing, truly amazing. I think everyone agrees with me. So apparently we are able to ask questions till about 3 a.m. Um, there are mics in the back, but we also encourage you to try some new technology and scan this QR code in your phone, and it'll take you to a Menti app where if you type in the code, you can type in your questions there. And this is also where you can access the survey for this incredible prize. So everybody, pull out your cell phones now.
There you go.
I-
If you don't want your ABIC swag, I will take it happily.
So I have to say I was inspired.
Oh,
and
good.
exactly what you wanted to do. It makes me worry about whether or not we are going to be denied access to these technologies. And I know there's a few other ag communicators in the room, Clinton, Monchuk, for example. Should we be targeting a specific group to have more impact? Should we be in the schools? Should we be in the legislature?
I think you have to be in social media. I hate saying it, but the bottom line is that this is where misinformation is thriving and where most people are turning for their information. And when you look at all the surveys, it isn't that people are against it or for it. They don't know what to believe. And just by being present in that space and commenting on what the realities are, maybe starts to leave a popcorn trail for people to be led in the right direction towards better discovery. Because you can't change people's minds, but you can really help them change their own mind. And I think that's really where we have a prominent role.
So I absolutely can't see the back.
I know.
So if there's anybody with questions, you're going to have to yell them.
I only see 4 people here. Yeah.
So going back to questions, I feel like super inspired. I may get a DNA tattoo, and I'm scared of needles. Noodles. So effective communication. I think that we actually should be designing our prize around who is using the hashtag and actually out there promoting this. And that's the way I think our next gift thing is going to go. Amy will make that happen. Amy makes all these things happen.
Well, here you go. There's a question.
All right.
Right? In the ag biotech industry, how do I answer the question, what about the long-term effects of genetic engineering? And I think the easy answer is that what we're doing in genetic engineering in plants and animal systems is really nothing different than what's been happening during evolution. That changes are occurring all the time because of errors that are made during DNA replication, during energetic events like cosmic rays from space or radiation emission from the Earth. Genetic changes are happening all the time. That's why we all look different and why we have so much variation. Genetic change is part of life. It's done in ways that are random. It's done in ways that we can't predict and affects genes that we can't control, leading sometimes to those types of diseases I talked about. When we're doing genetic engineering, we're making very precise changes that we understand, in genes that we understand. We're able to monitor their effects and test for their effects with great power. That's why I don't like using the word genetic modification. I use genetic engineering because we're using math and science to predict how this will work. As it was said, would you rather drive across an engineered bridge or a modified bridge? Right? There's power in that language. So this is engineering. And so when you're talking to people about it, saying that we're using hypothesis-driven science to make changes that we understand, and then we're testing to make sure they're safe. That's why this is such a powerful technology. Oh, wow.
Yeah.
There's more. Okay.
There are tons up there now.
Okay.
So the next one was, how do you approach concerns that biotech is playing God?
Well, because it's really not. I mean, and especially if someone says playing God, it may say that they have some religious concerns about tampering with nature. But humans have been tampering with nature for a long time, whether it was the plane that got me here or the vaccine that keeps me safe on it. We go against nature all the time. Nature, by nature, wants us to live till 20 years old, reproduce maybe once or twice, and then go away. And that we fought that back as a species by implementing more science. And so I don't know that it's playing God as much as it would be equally fair to say that it is using tools that God gave us to use to solve the human problems in the human condition. And I don't know which one that is. And maybe I'll get to ask him someday. But right now, all I can really do is we do the best with what we have. And if it's solving problems for the people in need, then that's what we need to be doing.
This next question is, we've seen all these great things that have come about. Do you have an example of a failed biotech that we may not have heard about?
Oh, sure, yeah. No, there's lots of them. The thing about the failed ones is you don't really hear about them very much. But one of the most spectacular failures was trying to make animal feed that was supplemented with an amino acid, I can't remember, named methionine. And this is something that's very helpful to have in animal feed. And they took the gene from Brazil nut and put that gene into corn to produce this high methionine feed. And it produced a protein which is highly allergenic in humans. So they ditched that project. The Flavor Saver tomato was the first genetically engineered food product that was approved for human use. It was supposed to make a tomato last longer and taste better, like it was picked from a plant ripe. The problem is they used a lousy tomato to begin with, so it wasn't any better than what you bought in the store. I mean, I can tell you over and over again about the things that don't work. In medicine, many examples of ways in which things don't work. The viral correction of genes is very dependent upon where they're delivered and if they are able to make that DNA transfer into those cells. 97% don't work. What it really says by saying there are so many failures isn't that it's sloppy or problematic. What it says is that we're very good at identifying the way to use it correctly and regulate it correctly, and that the things that don't make it to market have obviously been tested ad nauseum to eventually lead to their failure. You can be very confident that things that come through are with much certainty very reasonable and safe.
So is this the next scientific generation's race to the moon? Is the gene the next frontier?
Well, I'd like to say we're already at the gene, and I think when we sequenced the human genome and had a parts list for the human in 1999, we started to really understand we had the parts of what we're made of, and now it's a question of how these parts fit together and how they work together and what happens when a part is broken. These are the things we're learning now when we study genetics. The next generation's biggest task will be on multiple levels. How do we feed 10 billion people by 2050 with fewer resources? We have to use less fertilizer. We have to use less land. We have to use less water, yet we have to feed more people. And it's going to be huge innovations in genetics and in the way we farm, using all the really cool new tools of robotics and sensors and satellites and drones and electronics and data. This is what's going to drive the innovation that's going to allow us to feed the world with less impact on the planet. That's one major grand challenge. The other one is, as we start to defeat things like infectious diseases and heart disease, and maybe start to make headway in some of the more mundane ways in which have always killed people, we start seeing the onset of more long-term debilitating degenerative disease, things like cancers, neurodegenerative disease, Alzheimer's, Parkinson's. All of these become now the next wave of major issues we'll have to wrestle with. And these will be an incredibly challenging nut to crack for the next generation. That's the next generation's moonshot. How do we solve medical problems that will be the major frontier that's plaguing us and our healthcare systems? I think that's going to be extremely challenging. Those are really the big ones. How do we make more with less, and how do we solve the problems that will plague us as we're making more with less and living longer?
These are really good questions. We
Oh, I
should
love
have,
it.
we
This
should
is
have
great.
prizes for questions, right?
No, I absolutely love this.
So besides communication, do you worry about any specific developments within biotech, whether the science or the industry?
Yeah, so the big problem with this is that we've gotten biotechnology to the point where it is doable by just about anybody. And you can come to my lab, and in a month or two, I can have you schooled up on how to make recombinant DNA, bacteria. We can do that in a week. It's great tools, and they work wonderfully. The problem is just like anything else, there are people with malintent. And just as you can weaponize a 747 and fly it into a building, you can synthesize a new virus on a DNA synthesizer. And this is a very real threat, and something that I spoke at the State Department last month about, or 2 months ago about, and learned about. there are many, so it's not a question of how do we stop it from happening, how do we respond to it after it happens? And how do we identify the source, and how do we surveil for it? And how do we identify novel sources as they're happening? It's because these technologies have become so agile and so friendly that we have to start worrying about them being used incorrectly. And it's just like anything else, like a screwdriver, right? I mean, you can turn a screw or shove it up your nose, you know. I mean, it's any technology has its limits as to how it's used correctly. And I don't think that's a reason to put on the brakes. In fact, it's a reason for us to speed up. It's a reason for us to invest more money in detection and monitoring and understanding how these things can be done. So just like anything else, it does have its negative side.
So if you're a conspiracy theorist, this is a bad segue. Did the pandemic influence the public's view on genetic engineering?
I think it did, but I think it did it in a positive way. I think what the public saw is a viral disease occur that changed our lives. It impacted all of us in different ways, some of us in very profound ways where we lost family members. But the fact that we had a solution that came from laboratories within months is amazing to me. I mean, Jonas Salk and the polio vaccine took years to develop. You look at many of the other innovations that occurred in medicine took a long time to develop. Having new genetic technologies allows these things to be done rather quickly and still extremely safely. Not using the traditional way to make vaccines by inoculating eggs to make lots of virus and then purifying the proteins that would be used to confer immunity. That's a huge process that takes years of how do you check for safety and standardization and purification and inactivation of a virus. All of those steps have flaws. The mRNA manufacturing process really is one step. Putting that into a lipid nanoparticle is another step. But these are pretty simple and straightforward. So this is what's so exciting about these technologies. They give us the agility to move into new technologies, to address new problems. One additional thought on the lipid nanoparticle, this little bubble of fat that they put the RNA in. There are companies now that are making custom ones that can go to the prostate gland to deliver a therapeutic and shut down cancer and prostate cancer. That's very real and in clinical trials. New ones that have little bits of viruses on the lipid nanoparticle itself so that when it sticks to a cell, it fuses better and delivers more efficiently. It can go to places like the heart, the brain, the liver. You can target specific places with that particle. So it's not just the therapies, it's the way in which they're being delivered. Super cool.
I'm going to jump then one question over because it's near and dear to my heart. How close are we to dramatically increasing our lifespans?
Yeah, so the question, that's a tough one to crack. So one of the things that we're going to see almost with some great certainty is that diseases like cancers, and when you say, there will never be a cure for cancer because cancer is not one thing. But there's prostate cancer, and there's breast cancer, and there's different brain cancers. There's specific kinds of cancer that each can be individually addressed because each have their specific vulnerabilities if we can get therapies to them. And there are many in the area of cancer therapeutics that believe we may not be able to cure certain cancers, but most of them will be managed, meaning you'll take a pill every day or receive some sort of a shot once a week, that you will live with it. It won't kill you. It won't progress. It'll go into a remission. And this is seemingly something that gives me a lot of hope. I think that's something that will help improve lifespan. But something's always going to get you. And to me, I think about the other degenerative diseases that are really tough to crack, like Alzheimer's. The therapeutics, there's only 2 therapeutics on the market for Alzheimer's right now. Neither of them they're both on like emergency tests because they're very difficult to do clinical trials in Alzheimer's because people who are being tested already have the presentation of an advanced state of the disease. It's very hard to catch it early when it starts because you don't know people who have it when it starts. The early onset types are relatively rare. So those are going to be much harder nuts to crack, but it is estimated that children that are born today will routinely live over 100 years. My daughter will be on Willard Scott. If he didn't live that long, did he? Anyway, tangent.
How, how do you view corporate incentives, especially big pharma and big ag, and how they affect and influence research?
Yeah, so this is always a good question. You know, the problem with research that's corporately funded is someone's got to pay for research. And it isn't coming from our governments like it used to, and it isn't coming from our national organizations or from charities. Someone's got to fund it. And it's sad because so many great ideas are on the shelves at universities, some of them in my lab, because I can't get funding to pursue them. And that's just where we are. When corporate money is used to drive research, people automatically look sideways at it, that it's somehow tainted and problematic. But I think what's important to remember is that a company doesn't fund research unless they have a pretty damn good idea that it's going to work in the first place. So, you know, like folks like Marion Nestle would say, well, 85% of corporate funded research finds in favor of the company. That's true. But that's because a company doesn't go to a university and say, hey, Folta, I want you to check this out and see if you think it works. What it really is we have a lot of data that we understand this very well. Here's the experiments we want you to perform and see how it works in your hands. It's independent replication that we would do, and if it doesn't work, that's the end of it. You don't hear about it anymore. If it does work, we publish it. And now it looks like we agree with the company. If we don't agree with the company, nobody knows. That's the problem. You know, back to the drawing board, your stuff sucks, you know, get out of here. You know, so we don't necessarily, so that's why it always is tricky to talk about company research. But they have the money to be able to drive innovation. And to be honest, when I got approached with using one of our technologies and having some funding 1 year to have a scientist on the project, I was glad to take it because I think it allowed me to hire a scientist who I wouldn't be able to hire usually. And we had 1 year of research that was funded by Bayer, and it was fantastic that we were able to do it. Unfortunately, they bought a company and gave up the program that we were funded through. And so we were never able to finish that work. It's still pending. I should pass a hat around if you want to. Good.
After my hat.
That's right.
This is a tough question, too. Do you think there is a role for Indigenous knowledge to act alongside Western science in the development of biotech?
This is a tricky one for me because I don't ever like to be disrespectful to what people refer to as Indigenous knowledge. But what I do have is a very high bar for evidence. And I understand that people have certain feelings about you know, what we call Indigenous knowledge or tradition, that it worked before, apparently. We must have rules in science that say reproducibility and blind tests, hypothesis driven, using the best experiments and the most sensitive tools. And from that comes the best evidence. And it's great to use Indigenous knowledge as a hypothesis generator. This is what we always believed. Let's put it to the test. But it's hard for me as a scientist who must play within the bounds of hypothesis driven evidence to automatically accept something just because someone believes it. It's welcome to the table. It's welcome to be tested. We'll see if it passes the most rigorous tests. But that's always the way that I have to view it. I think that's really important. And that's a tough topic. It really is.
It is. How do we get more scientists to speak up like you do and talk about these things to consumers?
Well, you first have to find some people who are really excited to get abuse online. No, I say that semi-jokingly. And the reason I say that is because, you know, I gave this talk here today, and some of you will go home and Google me and say, you know, and see, you know, what a scumbag he is, and there's one big Pinocchio nose. And the bottom line is that whenever you say something that is exciting and forward-thinking, there's going to be people who don't like it. You know, there are people who are mad about the car because it was going to end the buggy whip industry. You know, you're always going to be stepping on someone's toes with progress, always. And so how do you get more scientists to communicate? The best thing is, first, arm them with the proper way to communicate. Teach them the ways that work and that fail when we talk to public audiences. The other thing we need to do is have more support from our institutions. We need to be rewarding scientists who get out and do this, not penalizing them. And scientists who step into the public discussion, you don't get gold stars and credit towards tenure and promotion. If anything, your boss takes you aside and says, this isn't really your job. It's a little bit of a distraction from what you're normally doing. And we need to change that. We need to be rewarding scientists who step into these conversations. And that's one way that we can really incentivize it. Most of all, I think what we have to do is indoctrinate a generation. I hate saying that word because in my state it means something else. We have to get a generation excited about the idea of sharing science and making it part of what they do. And I think we're getting there. I think that the people who I work with, they tell me I'm wasting my time. Why are you going all the way to Saskatoon? You know? And that's how it is, seriously. You're wasting your time talking to people about science. Nobody really cares. Folks in my classes, they get it. They see that if these kinds of innovations are going to become reality, they need to be out there talking about them and sharing them in social media.
This is another great question. How do we deal with selective acceptance? I am guilty of this. So we'll accept medical technologies. We might accept vaccines. Some vaccines are okay, some aren't. But GM crops and food additives, not a chance.
Yeah. So how do you say we're going to look at this through one lens of evidence-based decisions, and these we take, these we don't? And I think it boils down to the very complexities of humans and where we get our information and what we respond to. Who are the people who are helping to influence our decisions? And really what it calls us to do is it forces us to do two things. Test ourselves for self-deception. Check our own biases. Approach these questions with an open mind. Be able to seek information, but identify good information and bad information. Tell the difference between them. This kind of idea has really changed the way I work as a teacher. I used to cram my students full of facts and figures and biochemical pathways, and you got to know where all this stuff is. Now I don't do that. They know where to find it. They could go on their smartphone. It's all right in there. But how do they tell what's good versus what's bad? How do they look at it with a critical eye? And so the problem of selective acceptance is really a problem of people haven't been brought to the right evidence yet, or haven't been able to tell the good evidence from the bad evidence that's shaping their opinion. So if you see someone who's doing that, help lead them to some possible alternatives that may shape them in the more evidence-based direction. That's hard to do. People don't like changing their minds about anything. But it's not, and as I mentioned earlier, it's not about changing someone's mind. It's about giving them the tools and the incentive to change their own mind.
Which also segues into Europe. These Europeans.
Oui.
Is there growing acceptance for biotech in Europe?
Oh man, one of the saddest things is I have so many colleagues in Europe who are just so upset that the innovations their labs create are destined for the garbage can. They will not likely be accepted in Europe anytime in the near future, except in England, which is still a part of Europe, but not the EU. So that's nice for them. The European government is making decisions that are based essentially on politics, which are being disseminated and determined by the citizenry that doesn't like new technology. And the thing that's killing them, this technology where you can erase one letter or change one letter, this is so important in crops, and it still is illegal. Or the European Court of Justice said, we will not allow this in the EU. EU member states disagree wholeheartedly, some of them. And so it really is a question of it's going to change, but it's going to take some time. And the sad part is EU and EU science will be far behind. Right now, China invested $7 billion in these technologies. And they're dumping money into this. The writing's on the wall. They got a population to feed and limited resources. They see how it can work. In the US, heavy investment in these areas, but not the EU.
And I am concerned about Canada, I have to admit. And this next question actually speaks to that quite a bit. There's a movement for all natural solutions when talking about environmental sustainability. And there's a lot of controversy around GMO and gene editing. What would you say to this?
Yeah, that's kind of sad because, as I mentioned earlier, genetic change is natural. And being able to control that rather than leave it to randomness is really an important part of being able to move forward with sustainability. When you look at even natural solutions, what does that mean? It doesn't mean much anymore. Natural solutions are, until the last 100 years, one of the biggest problems in humans was malnutrition. The majority of humans were undernourished. And even in the United States, until after World War II, we had a lot of malnutrition in our country, in the U.S. So natural doesn't always cut it. And whether you're flying in a plane or taking a vitamin pill or on your smartphone, we defy nature all the time. And it's just okay. It's okay to defy nature because we're doing it to improve the human condition and hopefully improve our planet. We've done a bad job wrecking it. And we've taken on some bad behaviors as humans. And we should have all tools on the table to try to correct those things.
You mentioned Alzheimer's earlier. So what is your view on prion diseases and how would biotech influence research and treatment?
Prion disease. Who asked about prion disease? You're
Can't
awesome.
see them anyway.
Yeah, so prion diseases are really interesting. So like mad cow disease is a good example. These are an example. We have a protein in our body in all of our nerve cells called the prion protein. And the prion protein in its normal shape is doing some sort of role, and we're not exactly sure what it is. If you remove it from a mouse, take the book off the shelf and have a mouse without a prion protein, the mouse has variation in the way it runs on a wheel at night versus during the day. It's a very different, very minor disturbance in circadian rhythms. The prion protein, we don't know what it does, but what the prion protein does is it is influenced in shape by other proteins, by other prion proteins. So if a prion protein that's misfolded gets into you, it now misfolds your prion proteins, and they become pathogenic. This is weird. It's not viruses, it's not bacteria, it's not chemistry or environment. It is exposure to a misfolded prion protein that causes misfolded prion proteins. And in the area of mad cow disease, it was likely that these cows were fed illegally material from the brains and central nervous systems of sheep that had a disease called scrapie. It was a neurological disease. And this is what imparted the problem to cows. Prion diseases were first identified in humans in New Guinea, where there was cannibalism, where people who would eat the brains of someone who was deceased, which they usually give to the women and children to make them smarter, right? They would develop something called kuru, which was a prion-based disease. And this is in humans, it's rare still that you have prion-based disease, Creutzfeldt-Jakob, fatal familial insomnia. There's a number of diseases that are caused by this. So the question was back, how does biotech influence it? The thing about biotech with prion disease is probably not going to cure it. It may offer some therapies that can help you live longer and manage prion-based diseases, but it may be very powerful in the early detection of prion-based diseases by identifying misfolded protein variants either in potential food items to make sure it doesn't happen or in people who may have been exposed. Deer with chronic wasting disease is a prion-based disease, and it's passed in urine and stools in deer. So this is a really weird variant of this. You don't need to be exposed to the central nervous system to catch the disease in those deer. There has not been deer-to-human transmission documented, but certainly within cervids, deer to moose, deer to elk has been. So very interesting stuff in prions. It'll be interesting to see how biotech plays a role.
So sticking to the techie questions for a bit, is there some solutions for citrus greening or Dutch elm disease?
Yeah, citrus greening disease. Since 2003, the citrus crop in Florida has been dying. We had acres and acres of juice oranges, 60 million trees, and we're down probably to 15-20% of that. And groves that have been there for generations are now closing. The trees are dead. They tear them out, make a subdivision. We're losing that in Florida, which is really sad. It's an iconic part of our state. It's on our license plate. It's like if Saskatchewan got rid of wheat, and no progress on the disease. It's a bacterium that's spread by an insect And the best thing that we have are new types of citrus, which are more resistant to the disease, new varieties that have come through natural breeding, new roots that we can graft onto, which impart resistance to the top part. That's coming. There are some genetic engineering solutions, but Coke and Pepsi, which are Minute Maid and Tropicana, this said the consumers will not accept GMO orange juice or genetically engineered orange juice, so we don't even want you to go there. If we were allowed to go there back in 2005, I bet we'd have a solution today.
I think you should read this one with me, the long ones.
All right.
So would you say that the benefits outweigh the risks? For example, 50% relative frequency of off-target effects due to inaccurate modification of gene sites.
Yeah.
How would you relate that to ethics, and what is the boundary that we must not cross?
Yeah, so this is a really nice question. Thank you for this one. So when we talk about this idea of gene editing, being able to change a single letter, such as in, if we take well, it wasn't the case of Victoria Gray, but some of these other cases where we can fix one letter and then solve a problem, that is not a perfect process. So we have what are called off-target effects, and they can be frequent. Here it says 50% relative frequency. In some cases, they're much higher. And what it means is you make the correction here, but that means you make an error somewhere else. The good part about this is that we can sequence a genome in an afternoon for $1,000 and get all those 3.6 billion letters and then test to see where those mistakes are. And if they're in an area of the genome where there's no genes or information, that's pretty safe to say that there's probably no risk or little risk. And as long as the thing we're trying to solve is there, it really is a question of risk versus benefit. And if we do enough of these events, we can eventually find those where only the correct edit was made. We have some plants that we've done where we have only the target edit and nothing else. Really cool. But what's the boundary that we must not cross? You know, and that's really depends upon each individual case. And that's why these things have to be assessed stringently and individually. There are no two cases that are exactly alike. And so the ethics really lie in the researcher and the product. And I know that gives people a lot of chills, especially when there's money involved. But I think this is really where we're at. How do we regulate and not stifle a wonderful new technology? And this is a challenge. Someone asked about challenges for the next generation and the moonshot. The folks who want to get involved in ethics and law should have a real field day here. I mean, I think it's fantastic ground to really have some great conversations, and I would be remiss to say I have all the answers. I certainly believe that we're trying to do the right thing, and as long as our ethics are guiding us, then we'll be okay.
So has the non-GMO butterfly label damaged the egg industry?
I think it's given the false perception that there's something magical and safe about that are not non-genetically engineered. And the sad part about this is the way in which it's misused. I think it just causes more confusion. That when you have, you go and you buy some hummus and it's chickpeas and sesame oil and says non-GMO project, you go, yeah, but there's no genetically engineered chickpeas or sesame seeds. So there's no way anything in there could be genetically engineered. Yet it's got a non-GMO project label. And what it does is it implies some sort of special risk where absolutely none exists. And then when we do look at products that are genetically engineered, if you look at the genetically engineered crops are corn, soy, canola, and the other one is sugar beets are the things that we commonly are receiving in our diet. And it's all the ingredients. It's the starch, the oil. Soybean oil, you can look at it all day. You won't find any DNA or protein in there. It's oil. And so even though it comes from a genetically engineered plant, the ingredient is exactly the same as the one comes from a non-genetically engineered plant or an organically grown plant. Exactly the same. Canola from genetically engineered, not genetically engineered or organically grown, exactly the same. The difference is the cost to produce it. And so if anything, genetic engineering makes plants that are more efficient for the farmer, which means a lower price for the consumer. And for me, it's about access. It's allowing the poorest people to still have access to good food. And if tweaking a gene in the parent plant means the product that comes from it is more readily available. That's something I get very excited about.
I had to read this question a few times to understand it. So it was it, why are all the crazy anti-science people in California? Because I would argue there's a few in Alberta. But I think it's, is there a risk that that state is going to regulate genetics and biotech into oblivion?
I don't think so. You know, the issue with California, you know, they've got bigger problems. One is which they have no water to perform their agriculture anymore. They've got quite a few problems in California. And I don't necessarily I have trouble with calling people crazy as much as we haven't yet helped them learn what's going on. And they have concerns. And their sources have taught them that this is a problem, and in their hearts they believe it. And the first thing for us to do is not alienate them, but kind of welcome them under the tent and say, how can I help you understand this better? And that's the mistake we've made in science communication, is we beat them over the head with facts and evidence and expected them to change. But in their hearts, in the people they've talked to, have them convinced that it's a problem. The only way it changes is when we approach them softly and with respect and try to help them change themselves. So I don't know about California. I don't know which way that's going to go. I'm confident that the science will be so exciting, especially in medicine, that people will start to realize that this is a good thing. And they've been telling us that genetic engineering in crops is going to kill us all 30 years ago. And we're still here and we're doing okay. Some of us are reproducing. So it's pretty exciting that this technology still is here with us.
I like this question a lot too. So if you had $100 million, apparently we have to qualify it with Canadian, so it's not quite as good, but where would you invest
Oh, that's
it?
such a hard question to say. You know, 100 million. Is someone offering this? It's really tough. I don't know. I think that there are so many good companies right now, like CRISPR Therapeutics, the ones that are coming up with the cure that Victoria Gray received. They're the ones leading in the sickle cell question. Rocket Therapeutics is a great one, looking at they're publicly traded. They're looking at the rare diseases, and there are so many rare diseases that you can fix with this viral therapy that you could make a very good business model by correcting them all and by making it affordable. I think I would be really excited about investing in urban agriculture. How do we grow more with less in our cities and convert more space to efficient growing space, not instead of the farm, but in addition to? And I think that's a really exciting idea. I love that. Last time I was in Saskatoon, I got to go out to a place that was growing basil and lettuce inside basically one of these storage buildings. And it was fantastic, and they had a beautiful operation. The problem is right now the costs are so high for the energy. If we could come up with new ways and new plants to grow in them that are more energy efficient, I think we'd be in great shape.
I'm going to throw in one of my own questions. I don't know if you've read, one of my favorite books was Gwyneth Paltrow, Wrong About Everything. So when's your book coming out?
Oh, the problem with my book is I started writing it and I had a good start and I didn't start it. If Gwyneth Paltrow was wrong about everything, my book was about how was I wrong about everything and the mistakes that I made in learning how to be able to talk to people better as a scientist, and talked about what an arrogant jerk I was and how I was really bad at being the expert and telling everybody I was the expert, and not necessarily being kind and respectful about other people's concerns. And so my journey was a little bit different. And so my book was called Spectacular Failures in Science Communication and How to Not Make Them Too. And it got started, and then other stuff's got in the way, but it'll happen one of these years.
We want an advance order of 250 copies that we will, next time you come out, have you autograph. Not to disincentivize you to come, but I'm anxiously awaiting the book, and I cannot thank you enough for tonight's talk. We were talking about KISS coming to Saskatoon. They've got nothing on you. Technology rocks. Kevin Folta rocks. And join me in thanking our great speaker.
Thank you, everybody. Yeah, and reach out if I can ever be of assistance. I really mean that very honestly. Questions you may have, concerns, thoughts. If you want to co-host a podcast, if you want to help with some writing that you want to do, please reach out. It's part of the biggest joy is helping you get your voice in science.
I have to remind you about the survey. If you ate the popcorn, you will fill out the survey. And you're only helping us do a better job. Maybe we can next time put a question in an advance copy order of this book.